Hello, my name is Mia Stevens, and I'm a recent graduate from the College of Wooster, and I'm presenting work I did for my senior thesis, which is entitled "Hummingbirds Are Not Fabaceae Pollinators. Erythrina flabelliformis, a hummingbird pollinated desert perennial, has significant fine-scale genetic structure and low pollen movement distances." So first, let's talk about the idea of natural fragmentation through the form of the habitat sky islands. So sky islands is what we typically see over a series of mountain ranges, in that we have populations of plants existing on each mountain within the range that are isolated from one another through a geographical barrier. And so when we talk about natural fragmentation, we're talking about fragmentation that occurs over a larger scale than when we typically talk about anthropogenically caused fragmentation. So if we look at the Sierra Madre Mountains, we can see that we have these very homogeneous population, but then we also have these very isolated little pockets that would have little pockets of populations of plants. And so this begs the question of how are these populations of plants persisting through this isolation? How have they evolved to persist through the isolation from the other populations of plants? And so one possible explanation for this is that these populations are connected via pollen movement. And for if these populations were connected via pollen movement, we would see this reflected in the population's genetic structure. And genetic structure is the non-random distribution of alleles over space or time. And when what we mean by that is that you have clusters of alleles within or among populations of plants. Another factor along with pollen movement of genetic structure is dispersal. So we're talking about seed dispersal, but for the scope of this talk, I will be focusing on pollen movement. And an example in a study that shows pollen movement is in Kramer et al. 2011, and they studied two different species of penstemon. First, the one is bee pollinated, and the second one that is hummingbird pollinated. And so what we can see here is that each individual population of plants is shown with a square, and each color represents a unique genetic cluster. And so this just means that for each population in the bee pollinated penstemon, we have a unique color, which indicates that each population has its own unique cluster of alleles. And this just tells us that the bees are not moving because each population has its own unique genetic cluster or its own unique color. This tells us that pollen is not moving between populations of plants and that there is minimal gene flow between these populations. However, when we look at the hummingbird pollinated penstemon, we see a very different story. There is more of a smear of colors, and that this is telling us that these populations are not isolated from one another. However, the hummingbirds are creating gene flow and moving pollen between the populations of plants. But what this study isn't telling us is what's going on within the population of plants. And this is important because what's occurring within the population of plants isn't necessarily the same thing as what's recurring among populations of plants. And to answer that question, I'm going to introduce you to my dear friend, Erythrina flabelliformis. Erythrina flabelliformis is in the Fabaceae family, and it is hummingbird pollinated. Typically, we see black-chinned and broad-tailed hummingbirds. And Erythrina flabelliformis exists in this naturally fragmented environment of the sky islands. As you can see here in the Sierra Madres, we have a very homogeneous population, but then as we move further north, we have more isolated pockets of populations. And one of these pockets of populations is my study site at the Ameren Museum in Texas Canyon, Arizona. And as you might be able to tell in this panorama, the population is made up of clumps of granite rocks, and the plants only grow from underneath these clumps of rocks, so we end up with clumps of plants. The population total is around 500, but for my sanity's sake, I only focused on studying 120, and we chose this 120 by being within a specific geographical range within the population. And so my research objectives first were to determine flowering phenology over the season and if there's a correlation over space. and then see if this population exhibits fine-scale spatial genetic structure, and thirdly, to see if effective pollen movement distances is consistent with the fine-scale spatial genetic structure. And so first, a little bit about my field methods. As you can see here in this map, these are all of the different plants in the population. There were 120 individuals sampled by my collaborator and I, Lynn Lovelace, pictured here. And for 4 weeks in June, we collected count data on number of open flowers per plant, number of open inflorescences, and number of fruits. And we were able to actually determine which week the fruits were set by marking with a paint pen the highest open flower. And so what we can see here is this is my flowering phenology results. And on the x-axis, we have week or time. And then on the y-axis, we have number of open flowers per plant, and dot size is representing the number of flowers that an individual plant has. And the black square is the mean, and the blue rhombuses is the median. What we typically see with flowering phenology is that there's this highly peaked curve. And that's not what we're seeing here. We're seeing that there's high variation and a very non-normal distribution of flowering, and that there's just high variation in the number of flowers an individual plant has. However, if we look at flowering phenology over space, so what we have here is a map. We have longitude on the x and latitude on the y. Each dot is an individual plant, and dot size is the number of flowers per plant. And as the graph moves, the dot size is according to each week. And so what we can see here is that we're not seeing much spatial patterning, meaning that each cluster of plants is not flowering together. Flowering is occurring fairly synchronously. And so back to the research objectives. So flowering phenology was non-normally distributed with high variation and no spatial patterning. And so a little bit about my genetic methods. First, all of my genetic methods were based on a sample size of 127. The 127 differs from the 120 based on plants that we thought in the field were one, but actually genetically are distinct, or two or three. And so for each of these 127 plants, along with 43 offspring, I extracted DNA. I ran 8 different PCRs with microsatellite primers, and I ran fragment analysis to find the alleles of each individual. And then I used a program called Spaghetti to determine if the population shows genetic structure. And then I used a program called Service to find potential sires for each of the offspring. And so here we have my fine-scale spatial genetic structure results. On the x-axis, we have distance between plant pairs, and on the y-axis, we have kinship coefficient, R, and this just tells us how related two individual plants are. And so and then the red line is our observed, and the gray line is the 95% confidence interval. And so what we can see here is that plants that are spatially close to one another are significantly more related than we would expect. In fact, these points here that are around 2.5, these would be full siblings. And then if we go down here, what we see with plants that are spatially far distant from one another is that they're significantly less related than we would expect. And so what this tells us is that alleles within this population of plants are clustered, and that this population is exhibiting spatial genetic structure. And so yes, the answer to my second research objective is that alleles in this population are clustered. And so now a little bit more about my pollen movement distances methods. So we use the same 127 plants that we use for the spatial genetic structure. 14 of these plants were made focal plants. This just meaning that I collected 3 offspring for each focal plant, and focal plants are indicated in orange. And then I genotyped all of the offspring and found potential sires for each of the offspring, and then I was able to determine how far pollen was moved. And so what I'm going to show you next is what we would expect to see if mating was happening randomly. And when I say mating was happening randomly, I just mean that each individual plant within the population had an equal probability of mating with any other plant, and that there was no spatial patterning. And so what we have here is on the x-axis, we have distance between plant pairs in degrees, and proportion of plant pairs on the Y. And so what this tells us is that this just tells us how far, what proportion of plants are a set distance away from one another. So around 25% of the population is 0.000 degrees away from one another. Now we're going to add in what we observed. And so we have the same axes here, distance between plant pairs on the X and proportion of plant pairs on the Y. And what we can see here in orange is what we observed. And this is, we found significantly shorter pollen movement distances than what we would expect to see randomly. And we run the same analyses with a subset of these data that were assigned at a higher paternity confidence, and we found the same significance. And so is effective pollen movement distances consistent with the spatial genetic structure? Yes, we see that there was a significant pattern of short pollen movement distances, which is what we would expect to see with fine scale, with a population that exhibits fine scale spatial genetic structure. But to say anything with certainty, we need a larger sample size. And so some take-home points is that we found more fine scale spatial genetic structure than expected, and we found shorter pollen movement distances than expected. And so this could mean that these populations of plants are potentially more isolated than we thought. What we would expect to see if these populations of plants weren't isolated and that they were being connected through pollination is that we would expect to see low fine scale spatial genetic structure. However, since that is not the case, these populations of plants could be more isolated than we originally thought. I would like to thank my advisor, Dr. Jennifer Eisen, and my collaborator, Dr. Lynn Lovelace, and all of the members of the Eisen Lab who helped me with my genetic work. Feel free to contact me through email or Twitter if you have any questions. Thanks for listening. Bye-bye.